Jack the Ripper. Like, just hearing the name still gives people a shiver, even after all this time. He's the ultimate unsolved mystery, isn't he? London, 1888. A killer stalking the dark, foggy streets of Whitechapel. 5 brutal murders, maybe more, and a killer who vanished, leaving behind nothing but fear and a name that echoes through history. We still don't know who he was. People have been trying to figure it out for over 100 years. There are books, movies, theories, endless debates. Everyone wants to know. And one of the most fascinating ideas, one that pops up again and again, involves a piece of paper, a stamp, and the tiny bit of something left on it by the person who sealed it. We're talking about the From Hell letter. It's a truly chilling piece of evidence. This letter arrived with a small box. Inside that box was half a human kidney, preserved in alcohol. The letter said the sender had eaten the other half. Gruesome, right? This letter, because of that kidney, is often seen as one of the most likely to have actually come from Jack the Ripper himself. Most of the other Ripper letters are considered hoaxes, but this one feels different. It has a weight to it, and on that letter there was a stamp. A postage stamp. And stamps back then had to be licked to stick. Saliva. Human saliva. And saliva has DNA. So, the big question, the one that keeps people hoping, is this. Could the DNA from that postage stamp, all these years later, finally tell us who Jack the Ripper was? Could that tiny, invisible bit of spit be the key? It's a powerful thought. The idea of modern science reaching back into the past, pulling out a hidden truth. But it's not simple. Nothing about the Ripper case ever is. There are a lot of hopes, sure, but there are also a lot of hurdles, huge ones. Let's talk about that letter for a bit, because its story is pretty wild. It landed on the desk of George Lusk. He was the head of the Whitechapel Vigilance Committee. This group was formed by local residents, scared and fed up with the murders, trying to catch the killer when the police seemed to be getting nowhere. They were basically neighborhood watch, but way more intense. Lusk was a prominent figure, and the Ripper, if it was him, seemed to be taunting the very people trying to stop him. The letter itself was short. It was written in a rough, uneducated hand. It spoke of the murders of a plan to fry and eat the kidney. It was crude, terrifying. And then there was the package it came with. A small cardboard box, carefully wrapped. Inside, a jar, or maybe just some paper wrapping, holding half a human kidney. The letter explicitly stated it was from Catherine Eddowes, one of the Ripper's victims. She had a kidney removed during her autopsy, a detail not widely known at the time the letter arrived. This specific detail, along with the horrifying physical evidence, made the From Hell letter stand out from the hundreds of hoax letters that flooded the police and newspapers. It felt authentic in a way the others didn't. It felt personal. It felt like it was from the killer. After Lusk received it, he showed it to his committee members, then quickly took it to the police. It became a crucial piece of the investigation, though it didn't lead to an arrest. It was photographed, analyzed, debated, then, like much of the physical evidence from that time, it was passed around. It went from police hands, to archival hands, to museum hands, to historical researchers. It was studied, put on display, stored away, and every time someone touched it, every time it was exposed to light or air or humidity, something was happening to any tiny biological trace that might have been on it. That's a huge point when we talk about DNA. See, DNA is like a blueprint. It's the instruction manual for every living thing. And we all have our own unique version of it, mostly. It's what makes you you. When we talk about identifying someone with DNA, we're looking for these unique patterns. In forensic science, a tiny bit of skin cell, a drop of blood, a hair, or even a bit of saliva can leave enough DNA behind to build a profile. That profile is like a genetic fingerprint. It can point directly to one person. There are 2 main types of DNA we often talk about in old cases like this. One is nuclear DNA, or nDNA. This is the good stuff. It's found in the nucleus of most cells, and you get half from your mom and half from your dad. It's what gives you your truly unique genetic signature. If you can get a full nuclear DNA profile, you can often identify a specific person, or at least narrow it down very, very precisely. The other type is mitochondrial DNA, or mtDNA. This is found outside the nucleus, in tiny powerhouses within the cell called mitochondria. You only get mtDNA from your mother. So everyone in a maternal line has the same mtDNA. It's not as specific as nDNA. It can't point to one person directly unless that person is the only one in their maternal line, which is unlikely. But it's much more robust. There are many more copies of mtDNA in each cell than nDNA, which means it survives degradation better. So, for really old, degraded samples, mtDNA is often the only thing left. And here's the kicker: DNA, especially nDNA, is fragile. It breaks down. Over time, heat, moisture, light, chemicals, they all chip away at it. Imagine a really old, fragile book. The page is yellow, the binding crumbles, words fade. This DNA is like that. over 130 years, sitting on who knows what kind of shelves, handled by who knows how many people, that DNA is going to be in rough shape. People have actually tried this before, not just talking about it, but doing the science. In the early 2000s, there was a real push. DNA technology was getting good, and the hope was that it was finally good enough. Labs started looking at items from the Ripper case. The From Hell letter was definitely on the list. The researchers were excited. They carefully took tiny samples from the stamp. The idea was simple: extract any human DNA, build a profile, and then maybe, just maybe, compare it to the DNA of known suspects or look for matches in databases. But those early attempts, even with the best technology of the time, were largely a bust. Or at least, they didn't give the clear answers everyone hoped for. The main problem was exactly what we just talked about: degradation. The DNA was just too fragmented. It was like trying to piece together a puzzle where most of the pieces are missing, and the ones you have are faded and torn. They could sometimes get some DNA, often empty DNA, but it was usually incomplete or mixed. And then there was the huge issue of contamination. Imagine all those people who touched the letter over more than a century. Police officers, forensic scientists, back when forensic sciences was a very different thing. Archivists, historians, curious onlookers. Every time someone handled it without gloves, or even with old, less effective gloves, they could have left behind their own skin cells, their own sweat, their own DNA. The samples taken from the stamp didn't just contain one person's DNA, they contained a soup of many people's DNA. It was incredibly difficult to tell what was the Ripper's, what was George Lusk's, what was a police officer's, or what was a modern researcher's. Back then, distinguishing between them was almost impossible for such tiny, degraded samples. So, while the effort was real, the results were frustratingly inconclusive. It was a big disappointment for Ripper enthusiasts and scientists alike. It felt like the door was slammed shut. But science keeps moving, doesn't it? DNA technology today is a world away from what it was even 20 years ago. It's like comparing a clunky old flip phone to a brand new smartphone. The methods for extracting DNA have gotten so much better. Labs can now get usable DNA from samples that would have been impossible before. They have techniques that can separate incredibly tiny amounts of human DNA from other stuff, even from environmental contaminants. The ability to amplify DNA making millions of copies from just a few starting molecules is much more sensitive now. This process, called PCR, has been refined endlessly. It can work with much smaller, more degraded fragments of DNA. We also have techniques like whole genome sequencing, or WGS, which aims to read every single bit of a person's DNA. For old samples, this is still a huge challenge because you need a decent amount of good quality DNA, but the technology is improving constantly. More relevant for really old degraded stuff is something called SNP analysis single nucleotide polymorphism. These are tiny differences in our DNA that are common, but when you look at enough of them, they can create a very specific profile. It's a way to get information even from very broken up DNA, and then there's familial DNA searching. This is a game changer. If you can get a partial DNA profile, or even just an empty DNA profile, you can upload it to public genealogy databases. These are the sites people use to find their ancestors, to build family trees. And they contain millions of DNA profiles. If the Ripper's DNA is there, or if a close relative of his is in the database, the system can find a match. It won't tell you Jack the Ripper's John Smith. Instead, it might say, this DNA matches a third cousin of John Smith. Then, skilled genealogists can use traditional research, looking at family trees, birth records, death records, to work backward and build out the family tree, eventually narrowing down the possibilities to a small number of men who would have been alive in 1888. This is how some really old cold cases have been solved, including the Golden State Killer case. It's incredibly powerful, but it all starts with getting usable DNA. So, back to the stamp. Why is the stamp so special? Because of the saliva. When you lick a stamp, you're leaving behind more than just a little spit. Saliva contains cells, specifically buccal cells from the inside of your cheek. These cells have nuclei, and those nuclei contain nuclear DNA, so in theory, a stamp is a perfect little time capsule for a killer's DNA. It's a direct biological trace left by the person who sealed the envelope. How much saliva would have been there? Not a lot, probably. Just enough to moisten the adhesive. But even a tiny smear of saliva can have hundreds or thousands of cells. Each of those cells contains DNA. The key isn't the volume of saliva, but the number of cells and how well their DNA has survived. The composition of saliva itself is mostly water, but it also has enzymes, proteins, and those precious cells. The enzymes in bacteria and saliva can actually start to break down DNA pretty quickly, even without environmental factors. Yes, but if it dried out fast, and if the stamp was kept in relatively stable conditions, some DNA might still be intact. But Here's where we hit the giant brick wall, again time and environment, over 130 years. Think about it. That stamp wasn't stored in a sterile, temperature-controlled, humidity-controlled, light-free vault for its entire life. It was probably in a police evidence box, then an archive, maybe a museum display case under lights, then handled again. Each exposure to light, heat, moisture, or even just air degrades DNA. It breaks the long strands into smaller and smaller pieces. It damages the chemical bonds and makes it harder and harder for scientists to read the genetic code. Contamination is still the absolute biggest headache. It's not just the old contamination from 130 years ago, it's all the contamination up to today. Think of it. George Lusk received the letter. He probably handled it without gloves, his DNA could be on it, police officers handled it. Their DNA. Clerks, archivists, early researchers, modern researchers, every single person who touched that letter could have left a trace. Now, labs are incredibly careful. They wear multiple layers of gloves, full bodysuits, masks, work in super clean rooms. But none of that could prevent the contamination that happened before these modern protocols existed. If scientists try again today, they have to assume that any DNA they find will be a mix. How do you separate the Ripper's DNA from Lusk's, or from a police officer's, or from a museum curator's, or even from the person who put the letter in its current storage box last week? It's a colossal task. They might get multiple profiles, and how do you know which one belongs to the killer? You can try to rule people out. If you know who handled it recently, you can get their DNA and subtract it from the mixture. But you can't get DNA from every single person who touched it in 1888, obviously. And there's the amount of DNA. Was there enough saliva to begin with? Even if the Ripper had a lot of cells in his spit, after all this degradation, how many are left? It might be down to just a few barely detectable molecules, and even if they do detect something, what type of DNA will it be? Will it be enough nuclear DNA for a strong, unique profile, or will it just be mitochondrial DNA? If it's only empty DNA, it can only narrow down a maternal line, not point to one specific individual. It's useful, but far from conclusive on its own. Getting permission for a new test is another hurdle. Who actually owns the letter now? Is it a museum, a private collector? They'd have to agree to let scientists take samples. And taking samples often means a tiny bit of destructive testing, even if it's microscopic. Some historical artifacts are considered too precious to even risk that. There's an ethical debate: is solving the Ripper case worth potentially altering a historical artifact? Most would say yes, if done carefully and minimally. But it's a conversation that needs to happen. And then there's the cost. High-tech DNA analysis, especially for highly degraded and contaminated samples, is expensive. It requires specialized labs and experts. Who would fund it? Let's imagine the best-case scenario. Let's say, against all odds, a super-sensitive lab manages to extract a relatively clean, strong, nuclear DNA profile from the stamp. What then? First, they'd have to be absolutely 100% sure it's not contamination. This means a lot of careful testing, ruling out known handlers, comparing it to common contaminants. It would be a very cautious announcement. If they believe they have a genuine profile, then the next step would be comparing it. But comparing it to what? There's no Jack the Ripper DNA profile on file. This isn't a modern case where you check against a police database. So, the power of familial DNA searching comes into play. They would upload this profile, or even a partial one, to those public genealogy databases. If a match is found, even a distant cousin match, then the real work begins. Genealogists would start building family trees, looking for common ancestors, tracing lines down to men who would have been alive, and of an age to be the Ripper in 1888. This is how you start to narrow down stuff. It's a painstaking process, but it works. Imagine if they found a match to a known Ripper suspect, like Aaron Kosminski. Kosminski was a Polish immigrant, a barber, who was identified by a high-ranking police officer as the Ripper in his memoirs. He was supposedly put in an asylum for mental illness not long after the murder stopped. Recent DNA claims from a shawl supposedly found at a crime scene pointed to Kosminski, but those claims are highly disputed and often picked apart by experts because of huge issues with contamination and methodology. But if the stamped DNA pointed to him too, that would be a powerful, powerful piece of evidence. It wouldn't be absolute proof, because circumstantial evidence and interpretation still play a role, but it would be huge. But there's also the danger of misidentification. What if the DNA isn't the Ripper's, but someone else who happened to lick a stamp on the same letter? Or someone who touched the kidney, then touched the letter? What if a partial profile leads to a false positive or a misinterpretation? The stakes are incredibly high with the Ripper case, and any findings would be scrutinized to an insane degree. The scientific community and Ripperologists would tear it apart, looking for any flaw. So, Any conclusion would have to be incredibly robust. The From Hell letter stamp isn't the only piece of Ripper-related material that people have hoped to test. There's that shawl that recently caused a stir, although many experts have dismissed its DNA findings as unreliable due to contamination and improper handling over the years. There are other letters, though. Most are fakes. There are police records, artifacts from the victims. Every piece of physical evidence from that era is a potential treasure trove, but also a potential minefield of contamination and degradation. The Ripper mystery endures because it touches on fundamental human questions. Why do some people do such terrible things? Can justice ever truly be served, even after so long? There's a deep human need for answers, for closure. And the idea that science could finally provide that, could lift the veil on one of history's most terrifying figures, is incredibly compelling. It's why we keep talking about it. So, could DNA from the From Hell letter stamp identify Jack the Ripper? The honest answer is it's possible, but it's a very, very long shot. The challenges are immense. 130 years of degradation, a century of contamination, the tiny amount of original material, the difficulty of distinguishing one person's DNA from many. Modern DNA technology is incredibly powerful, far beyond what anyone imagined even a few decades ago. It offers a glimmer of hope. The techniques for working with degraded, contaminated samples are improving all the time. Familial DNA searching is a revolutionary tool. But even with all that, finding a clean, usable DNA profile that can be definitively linked to the killer, and only the killer, from that stamp would be nothing short of a miracle. It would require extraordinary care, advanced science, and perhaps a bit of luck. If it ever happens, if a robust, undeniable DNA profile emerges that points to a specific individual who matches the historical criteria, it would be one of the greatest forensic triumphs of all time. It would rewrite history. But until then, Jack the Ripper remains the ghost in the fog, his identity still hidden, perhaps forever. The hope is still there, though. A tiny, fragile hope, like a single, almost gone DNA strand, just waiting to be read.